Okay, hi. So I noticed on the schedule there that it said I have 10 minutes, but that's wrong. It's 20, right? Okay, thanks. So hello, everybody. So this is a nice opportunity to follow up on Victor's talk, actually. So maybe it can be, in a sense, well, if not interactive, then you can maybe think about what you just heard and apply what you learned if it was new information, because there's specific decisions that we made for the task-based stuff that I'm about to show you, where we had to choose which sequences we want to use, and there's trade-offs. And we have to decide, as Victor said nicely, about what our clever paradigm will be to try to get around some of these problems that you have with the different trade-offs that you make. But so what I'm going to be talking about is a layer-resolved experiment that we did in the inferior frontal cortex, which is a bit unique because normally people look at more sensory cortices when they do this. But so this is a reading experiment focusing specifically on Brodmann area 44, the dorsal section. So left inferior frontal cortex, very language important region, not just language, other things too, but it comes up a lot in the language literature. There has been some work, and I get the sense that the field is moving in this direction to do layer-resolved imaging and cognitive neuroscience in non-sensory cortices. So Emily Finn has a paper where they looked at working memory. I know that that's becoming more popular to do now, also in prefrontal cortex. The previous experiment that I did that this one is based on, or that the data came from anyway, was focusing on the left posterior temporal cortex and also what some might call the visual word form area, but a portion of the left ventral occipitotemporal cortex important for reading. So we're moving in this direction, but in the previous experiment that I did with the data collected at the Hahn Institute here, we never looked at the prefrontal cortex. So it was the next logical step, and I think a very exciting one, and I'm happy to be able to talk about it with you guys today. Yeah, so this is, in a sense, the requisite layer slide, but I won't spend too much time on it. But so there is a layered structure in the, well, not specifically in the isocortex, but that's the one we're talking about, the 6-layered structure. So you may hear neocortex, or you may hear it referred to as eulaminate cortex, but people are describing variations on a theme of portions of the brain that have this 6-layered structure. And you can see it's not always the same. Well, if you look on the left, your right side, your left side, you see BA 44 and 45. So these are two important areas for language and other things as well, but I talk about them in the language context. And you see they have this 6-layered structure, but how the 6 layers are distributed isn't always the same, right? There's differences in cell types in different regions. Exactly how thick the layer 4 is a big one. But you generally have this 6-layered structure. And importantly, it has functional implications. So the canonical microcircuit is an oft-discussed concept that we use very heavily, or that I make use of very much to generate predictions about what I think different layers would be doing in the context of a task. And you see that you have, in the bluish arrows, you have these forward connections. So these would be from region. So if you imagine this collection of circles here as a brain region, imagine there's a region that is by the forward connections arrow. So those are lower order input to this region, right? And then, so these backward connections, these would be what you would think of as maybe top-down or feedback. Top-down's a bit strange to use because people often mean it in cognitive terms, and sometimes people mean it in anatomical terms, but the backward connections. And then you have this whole mess of these intrinsic connections too, which are going to be important for later, so do make note of them, that you have this loop going from the more superficial layers to the deeper layers, and then you have these sort of relay loops going through the layers from the deep to the superficial. And this is an incomplete picture, but this is the basis of the canonical microcircuit. And like most of these things, it's almost certainly wrong, but it's right enough, we think, for certain fMRI experiments that we do when we have typically poor resolution compared to the actual histology that we're measuring. Yeah, so for those who are acquisition protocol minded, these are two of the important sequences that we used. So we did a gradient echo 3D EPI with sub-millimeter resolution, 0.9. And then we also had, importantly, a distortion-matched inversion recovery EPI. So a problem that you have when you do this type of work is that you need to get 2 brains into register, which are not the same, right? And because they're not the same, they'll never be the same. So it's a big challenge to solve this problem. So typically people do distortion correction and things like this. But we opted to actually collect a T1 weighted data set that was going to be distortion matched to the T2 star weighted data set. So that was how we went about this. So we have our, you can see under the gradient echo column, you can see our functional data. That's an example of one subject. And then you can see an example of the inversion recovery EPI. So you see the T1 contrast, but you also see it's distorted. It's an EPI image, and that's clear. And the red blob is part of the visual word form area that we identified in the previous experiment. But so it's not strictly relevant for this study, but you can see an idea of the two images that we would get. Yeah, so now I'm going to talk about the paradigm that we used in the previous experiment and also was the basis for the analysis that's coming up. So this was a reading experiment, and people would be placed in the scanner, and they would read these single words. And the single words it was all in Dutch, and they would be either real words like salmon or battery, pseudo words, which, so the idea about pseudo words is that they can be legal Dutch words, if they just happen to have meaning. But these are words that obey the sound patterns in Dutch, obey the rules in Dutch to be words, right? But they're just not. You could make up a meaning. And then the false fonts are kind of a baseline condition, and in certain senses it's a baseline because they conserve a lot of the angular properties of the Latin script, and they're completely devoid of orthographic meaning. They can't be pronounced, they can't be understood. But importantly, there's also, we conserve the statistical regularities about like n-gram frequency with these so that they match a lot of the low-level properties of the pseudo words. So you can see this kind of gradient where you have real words with meaning, words that could be real with no meaning, and then the false fonts. And then we had this block design type experiment where people would see a mini block of the real words, a mini block of the pseudo words, a mini block of the false fonts. These would be pseudo-randomized. But one thing also that's important to keep in mind is that there's this length manipulation as well. And that's going to come up in a couple of slides. So yeah. Now, how do we actually extract the layer signal? So that was the experiment. People would just be reading the scanner, but then we need to get the signal out. And this is a slide to show you just a summary of what that process might look like. So if you start on your right side, you can see on the top left of that, in the green box, this is an example of the ROI that we used for identifying the reading region. And if you zoom in, you can see what the surfaces actually look like on functional data. So we would generate the surfaces on the T1 weighted API using FreeSurfer and a lot of hand optimization of what goes wrong, right? And then you see you actually get very well, you get the surfaces very well aligned to the T2 star data. So you can see that in the second row here, what that actually looks like. And then if you put that back onto the T1 weighted image, you can see that the surfaces are actually pretty good, right? As far as you could tell with your eyes and according to the algorithms used. And so you have this nice definition of the surfaces, which corresponds to the reality that you can observe. And you can then draw surfaces, draw boundaries between these white matter and CSF surfaces. And when you do that, you end up with these different partitions, and you can have as many as you like. Often people have 10 or 12. We use 3 of them because we use a method to extract the signal, which works best with 3. It's the spatial GLM, which you can, which isn't specifically visualized here because it was in the overflow section. But the idea, once you have these boundaries, is that, so if you see on the bottom left, you can imagine that each of those colors of the rainbow, that would be one of the surfaces, the intermediate surfaces that we create. And then that black box, that would be a voxel. And you can see the fractional volume of every voxel is going to consist of the, some combination of the different surfaces that you have there. So you can imagine that if the voxel was all in the white area, that would be completely in CSF, beyond the blue boundary rather, in the white. And then if it was beyond the yellow boundary, you can imagine that would be entirely in white matter. But most voxels are a mix of these layers, right? So for a region like this, you would go through every voxel that you have and then look at which of the layers the voxel is in, and then you end up with a spatial design matrix, right, where you can have for layer 1, for all the voxels in your ROI, you have some percentage of the volume that's in that layer. and then you regress this against the signal and the ROI, and you do this for every time point, and then you end up with the time course for each of the layers, which is very cool, but not the subject of the talk. And so the previous results, we demonstrated that you could do this, and that we found interesting responses in the deep bin where, although the overall response showed that it was a stronger effect for pseudo words than real words, this wasn't uniform across the layers. There was something unique going on in each layer. But more importantly was that we showed that the deep bin specifically had connectivity to regions that are associated with lexical memory, different phonological processes. And this was unique to the deep bin, not the middle bin or the superficial bin, which was very cool and inspired what we would do now. Yeah, so most of what I just showed you had to do with low-level stuff comparatively, right? So we were interested now in moving to more prefrontal cortex. So the question is, what can we investigate in the inferior frontal cortex with a word-reading task? As maybe those who study language, this isn't very common to do. Usually people are doing some kind of, like, say, syntactic ambiguity or something with heavy semantic loading on it. But also articulation, covert articulation, processes that will maybe be driven by reading of, say, long pseudo words, for example. And that with the paradigm that we had before, you can imagine that reading words and reading pseudo words could engage these different processes. Perhaps with the absence of automaticity that you would get from reading real words, reading pseudo words would be more effortful, rely on different pathways, and very well might engage this more articulatory network that you see a popular version of a dual route model here that's showing a number of regions which are connected. And could we learn something about them by looking at how people process, phonologically difficult, unknown, novel pseudo words compared to real words into this baseline condition? And the other important thing is that, so the inferior frontal cortex isn't just this uniform blob. Right? There's some functional distribution of what's doing what within the region, and even that is probably hopelessly simple at this point. But there is a rough gradient of functional. It's not strictly necessary, but there's more involvement, say, more recruitment of different portions of the inferior frontal cortex for tasks that lie within different domains of language. So, say, phonological processing, for example, would be something that would be more dorsal, so BA44, the region that I'm talking about today. Whereas you have syntactic and semantic processing operationalized with different tasks, right? Because that's how we look at this stuff. But then you see you have this more focus in BA47, BA45. So there's this functional gradient there, which is why I thought it would be interesting to look at what would BA44, which is near that region that's labeled with a phonology arrow. How might that connect to these other regions? So I think if you look at the contrast at the non-layer level of just what the pseudo words compared to the false fonts give you, you see a very similar network emerging, right? So you've got this more ventral region that's probably related to the word reading, right? And then you have this posterior temporal region, you have this temporoparietal region, you've got these premotor regions and inferior frontal regions. So the network appears to be there. The idea that I had was to look at how the inferior frontal cortex was connected to these regions and what the layer properties might be. Because the layer properties of the connections would perhaps be able to tell you, is the signal that is relating these two regions, is that more related to input from lower order regions, or is this related to more top-down signaling coming into the region from higher order regions? And you should be able to see both, right? As a function of the task contrast, you should be able to see connectivity from inferior frontal cortex to more temporal regions, and from inferior frontal cortex to more sensory motor regions. If the idea behind the layers works, that different layers are related to input and related to output from different hierarchical origin points, then this should work. So I think I'll skip over the Y slide that I already skipped over, and I think I mentioned all of it incidentally anyway. Right, so what I did was I took from this contrast map, identified these 4 regions and then one seed region. So the seed region is the dorsal portion of BA44. And there's some specific reasons that it's interesting to look at this, but I think the most important one to mention for you guys today is that this is in some ways a practical limitation, because if you wanted to look at the ventral portion, so you can see underneath the yellow region, there's this sort of U shape there. It'd be very challenging to actually get the layers there, because when you have a nice flat region of cortex, then it's easy to know, well, where are the boundaries? And when you're deep into sulcus, it's very hard to tell. You have lots of partial voluming. And worst of all, if you cross the chasm of CSF there, you end up having superficial layer of one side matched with the superficial layer of the other side. So you're going to have enormous signal. It's going to have very little to do with what you want to measure. So it would be interesting to look at the dorsal versus ventral, but I selected the dorsal region. And then so we have these 4 other regions, which are predicted to interact with the inferior frontal cortex in a number of different theories, but for articulation as well, sensory motor planning, even though it was just reading. Wrap it up. Yeah. Okay. Right. So there are a number of different connections that are running through these regions. I will pass over this. But so yeah, here you can actually see what the layering looks like in this specific region. So we've got the deep, the middle, and the superficial bin represented there with green, reddish, and purple. You can see in the oval where the region was, and then on the right you can see what this region actually looked like when I identified voxels in it. You'll notice that they're not contiguous, and the reason for this is that there are some voxels that just didn't respond to the task as well, that had dropout, that were in regions with partial voluming, so you don't end up with this perfect region. But the region should be contiguous in the sense of functional specialization for this specific task. Right, so when we look at the responses to the real words and the pseudo words, so these are the layer responses in terms of percent signal change, with short and long, short and then long, all kind of broken out. And notice that they all increase towards the surface, and this is gradient echo bold, so this is in agreement with what Victor was saying. But you'll notice that there's some interesting differences, perhaps, like, so the long words have some differences in the convexity, concavity compared to the short words. And when you combine them, you see some residual portion of this in the plot. So while they're both increasing towards the surface, it appears that the actual difference between the conditions may not just reflect this staircase type shape that you see represented with the beta weights there. And for the false fonts, you get this much flatter response. So when you actually test for this, you do see that the response is higher in the middle bin. And this is stuff that we need to look into more, and there are different reasons why this could be, but tentatively it seems like the response difference between the conditions is driven by input from lower order regions, which would make sense if the inferior frontal cortex is related to lower order regions to help integrate over integrations in language, or just to deal with complex structures or sequence matching, these kinds of things. Skip this. Yeah, but the thing we really wanted to test, though, was the connectivity. And to do this, we used a tool called the psychophysiological interaction in the generalized form, where you have, you can see on the top left, you have the interaction between 2 regions. Normally, you can't say much about what's causing what, but when you have this layer information, you might be able to look at, say, hey, does this B region, does this relate to signal in the deep or superficial bin of a region, or does it relate to signal in the middle bin of the region? And then if you have a good idea of what your task is doing, then you can combine all this information together, and you can extend the generalized psychophysiological interaction analysis to give you the power to make inferences about directed connections, which is very cool. Then it becomes more like DCM, but I think a lot more wieldy. So I'll skip over all the model details. But so we did this in 2 ways. We looked at the connections within the region, so from one layer to another layer in the region, and then through the whole brain. And within the region, sorry for the quality of the plots, this was a new analysis. Within the region, there were connections that we saw between the deep and superficial bin as a function of the pseudo-minus false word contrast, which, if you remember back to the canonical microcircuit plot, there was this big looping arrow going between these 2. And also from the deep to the middle. So interesting interlaminar connectivity that corresponds with what we think about the canonical microcircuit. But the most interesting thing, of course, is this region to whole brain. And so on the bottom for reference, you can see this is my last slide, sorry, that so in the top left, if you take the difference between the middle and the deep bins, take the difference between the middle and the deep bins, just for the long pseudo words that showed the interesting difference in the middle bin, then you see that what is related to the signal in the inferior frontal cortex is this temporoparietal region, which according to this model and many others, should be sending input to this region. And then if you look at the difference between the middle and superficial, which I'm operationalizing here as either feedback or recurrent signaling between the two regions, then you see that it relates to these more like orofacial motor regions. So I think that there's a lot more to do with this, but it's very interesting that we can take the contrast of the different layers, apply these connectivity methods, and then start to see these maps emerging, which point to direct connections between regions in the brain that are interpretable, and then add a lot to the interpretation going forward of what the data mean for theories like this, and how we can think about dynamic networks that serve language. So yeah, that's all.